Hello, my name is Casey Holley. I work as a Claims Communication and Education Manager within our Research and Development team for personal care. Now before I begin, I need to set the stage a little bit. In the personal care industry, it is imperative that we separate product from the science. Products must be cosmetic, which is defined in most of our markets as something like intended to be applied to the human body for cleansing, beautifying, promoting attractiveness, or altering the appearance. Cosmetic products and their ingredients cannot claim any more than visibly improving skin. Yet, if we are simply going to talk about science and what it has discovered, we can talk about mechanisms of action. These could potentially extend to structure or function of the skin. Again, this should not include our products. So today, I have the pleasure of speaking on some of our science. We're going to focus on the research as it relates to targeting the sources of aging. In 2008, we announced our first foray into targeting the sources of aging with AR-NOX. Now, prior to this, Nu Skin research and development teams had been collaborating with researchers at Purdue University. This revealed a primary, previously hidden source of aging within each of us. The AR-NOX enzyme is an internal generator of free radicals. It is present on the surface of our cells, generates damaging free radicals, and it becomes more efficient as we age. AR-NOX is located on the external cell membrane and was identified within multiple sites, including both the dermis and the epidermis. Its unique property is that it generates superoxide, a potent free radical, at the cell surface that is capable of damaging adjacent cells, lipoproteins, and other structural components of the extracellular matrix, such as collagen and elastin. Present on the surface of our cells, it becomes more efficient at production of free radicals as we age. AR-NOX activity increases during the aging years of about 35 to 70. The activity level of AR-NOX is genetically dependent. The activity or inactivity of this enzyme may account for why some individuals look older than they really are, while others look younger. A study conducted at Stanford University has shown that individuals with low AR-NOX levels as a group are clinically graded to look an average of 7 years younger than their actual chronological age. Now, the group of individuals with high levels of AR-NOX appeared to be an average of 7 years older than their actual age. In additional breakthrough research, we scientists have identified a blend of botanical ingredients that appears to slow the production of free radicals in the skin. Specific botanical blends reduce the creation of free radicals associated with this AR-NOX enzyme. The inhibition of these free radicals may help slow the signs of aging potentially caused by accelerated free radical production. Unlike other free radicals that come from the environmental exposure, like the sun or pollutants, our genes control its activity. This directed our research to genetic fluences, as illustrated in 2009. Just prior to this, the International Research Project to determine the DNA sequence of the entire human genome completed in 2003. This decoded the approximate 30,000 genes in the human genome, laying foundation for additional research. The use of this knowledge and technology like gene chips provides a map of over 20,000 genes at a single moment. This is an immense amount of data, and it can be overwhelming. Yet our unique approach allows us to focus on a small set of robust biomarkers for rapid screening of phytochemicals and nutrients. DNA microarrays, gene chips, are like snapshots of what the genes are doing. Yet technology keeps advancing. PCR plates allow scientists to look at the gene's expression through time. This is more like a video compared to a photo of gene expression. In research collaborated again with scientists from Stanford University, this research identified 3 things: unique gene expression profile for normal skin aging, a novel gene expression signature for older individuals with skin youthfulness, and novel DNA methylation patterns for older individuals with skin youthfulness. These signatures can be used to test topical formulations and supplements for their ability to mimic youthful skin. Now, this understanding led us to the development of different blends of ingredients that can target functional groups of genes. We're not trying to change genes. Rather, we are either going to upregulate or downregulate their expression to mimic a more youthful gene expression profile. Here you can see a specific group of genes responsible for the structure and firming, hyperpigmentation, hydration, and cell turnover. Each of these characteristics is impacted as we age. Yet we don't focus on a single gene or even a single family of genes. Rather, our goal is to target this functional group. Further, we're not looking to simply upregulate, but bring them back into a balance. Upregulating genes responsible for the creation of proteins, while downregulating genes responsible for the degradation of these proteins is an example. At times, though, we are interested in a more narrow skin characteristic with the ingredients we are researching. Using firming as an example, which would be a subset of our structure functional group of genes, this specific focus is to target sagging skin and bring about a more youthful definition and contouring. Because it's related, you will see similarities in the types of gene expression being targeted. This blend was chosen as it can decrease the breakdown of structural proteins while also helping to promote and strengthen the extracellular matrix. You still see the rebalancing, the upregulating and the downregulating of genes for a more youthful gene expression profile as it relates to having firm skin. We have also looked at targeting genes that are related to youthful vibrancy. These genes are not so much about improving appearance, yet helping to mitigate aging and the maintenance of a youthful look or gene expression profile. Here we were focusing on genes associated with increasing antioxidant protection, keeping skin calm, and maintaining a vital moisture barrier. Here again you can see we are looking to upregulate and downregulate genes in corresponding directions to help skin keep looking youthful and vibrant. We want to increase antioxidants in the stress response but decrease inflammation or skin barrier disruptions. This will help keep skin looking its best through time. So what is unique about Nu Skin's approach? From the beginning, we didn't focus on a single gene or even one family of genes. We're not trying to just upregulate or just downregulate. Rather, we are focused on rebalancing gene expression. And then we confirm those benefits by developing final products that are put through clinical testing to ensure they have these benefits. We also share this scientific work with the community. You can see our science presentations that have been presented at meetings around the world. This research has resulted in numerous scientific posters, awards, oral presentations, and patents. When the research is presented at different scientific meetings, it's always well accepted. These are the numbers through the last year. Presenting and staying connected to the scientific community helps us to further the overall research and brings more ideas to our robust team of scientists. We are excited to see where the next developments in science will take us.